Hello everyone, this is Miss Hazy, and if you want to keep updated of my next lesson videos, please subscribe to my channel. Thank you so much. The following presentation will be all about the structure and functions of subcellular components, the organelles. Organelles, or the little organs of the cell, are very small, yet performs various functions for a cell. Mostly are found in the cytoplasm and may or may not be membrane bound. Talking about organelles, let's start with the outermost part of the cell, the cell wall. If you examine the picture, as you can see here, you will see the external part pointed by the black arrow. It is the cell wall. The cell wall is a rigid covering that protects the cells, provides structural support, and gives shape to the cell. Fungal and protestant cells also have cell walls, while the chief component of prokaryotic cell walls is peptidoglycan. The major organic molecule in the plant cell is cellulose. It is a polysaccharide made up of glucose units. Then we have the cell membrane or the plasma membrane, also known as the plasmalemma. It is a phospholipid bilayer with embedded proteins that separates the internal contents of the cell from its surrounding environment. The cell membrane is a semi-permeable membrane which only allows specific substances to enter and exit the cell. As presented, the yellow heads you see are the phospholipids forming up with tail-to-tail ends, a bilayer. A phospholipid is a lipid molecule with two fatty acid chains and a phosphate containing group. The hydrophilic or water-loving head of a phospholipid contains glycerol. Glycerol is a simple compound which is colorless, odorless, viscous liquid that is sweet-tasting and non-toxic. And the hydrophobic or water-fearing tails are made up of fatty acids. These phospholipids can actually move laterally or from side to side to allow small molecules such as oxygen gas, carbon dioxide, and water to enter. Molecules in the cell membranes are constantly changing and moving, and the movement is usually from side to side, as you can see in the illustration. Proteins in the cell membrane help move large molecules and aid in cell recognition. We have two types of proteins in the cell membrane, namely peripheral, or those attached on the surface, and integral proteins, or those which are embedded completely through the cell membrane, such as protein channel. Other examples of peripheral proteins are those involved in electron transport chain, such as cytochrome C, copridioxines, high potential iron protein, adrenoxine reductase, and many others, while integral proteins include insulin receptors, glycophorin, rhodopsin, cell adhesion proteins such as integrins, cadherins, selectrins, and many others. Next, we have the cytoplasm. The cytoplasm contains all the necessary life sustaining components. It consists of a thick semi fluid aggregate of chemical compounds called cytosol, various structures, and it also consists of internal membrane or the cytomembrane. The cytoplasm is composed of organic compounds, consists of proteins, fats or lipids, carbohydrates, and nucleic acids. The inorganic compounds, such as water, constitute the non-living matter, but still play important roles in living things. The cytoplasm is largely water. This is highly significant considering that water is known for its special properties, one of which is that of being a universal solvent. That is, water can dissolve more materials compared to many other solvents. Hence, the presence of many health sustaining substances in the cytoplasm. Likewise, protein is the most abundant among the organic components. of the cytoplasm, because protein provides materials for the growth and development of the cell. The cytoplasm serves as the reservoir for the entry and exit of materials in the cell. It provides turgor pressure. It is the site of many metabolic reactions because it has a network of membrane bound pathways for materials which the cell needs to sustain its life activities, and it is the medium in which organelles are found. And then we have the ribosomes, or the protein factories for the cell, where proteins are synthesized by joining amino acids from the coded messages from the DNA as template. Such message is carried by the messenger RNA from the nucleus to the ribosomes in the cytoplasm in the process of protein synthesis. Ribosomes are particularly abundant in cells that synthesize large amounts of protein. For example, the pancreas is responsible for creating several digestive enzymes, and the cells that produce these enzymes contain many ribosomes. Some ribosomes are bound or attached to the endoplasmic reticulum, while others are free or suspended in the cytosol. Bound or attached ribosomes make secretory proteins, while free ribosomes produce proteins that are localized in the cytosol. Next up, the mitochondria, which are often called the energy factories of a cell because they are responsible for making adenosine triphosphate, or ATP, which is the cell's main energy carrying molecule. Mitochondria are the sites of cellular respiration through which they harness energy by breaking down food molecules to make ATP. In mitochondria, this process uses oxygen and produces carbon dioxide as waste product. In fact, the carbon dioxide that you exhale with every breath comes from the cellular reactions that produce carbon dioxide as a byproduct. As you can see, mitochondria are oval-shaped, double membrane that have their own ribosomes and DNA. Each membrane is a phospholipid bilayer. The inner membrane, as you can see, is thrown into folds called cristae, which provide more surface area for greater cellular respiratory productivity. An example of the complementary of structure and function. The inner cavity into which the cristae project is called the matrix. It is filled with a semi-fluid medium containing enzymes that break down carbohydrate products. The narrow region between the inner and outer membrane is called the intermembrane space. Then we have peroxisomes. Peroxisomes are membrane-bound vesicles which contain oxidative enzymes. As you can see in the figure, its membrane is also made up of lipid bilayer, and it has a urate oxidase crystalline core. They have enzymes for oxidizing certain organic molecules, resulting in the formation of hydrogen peroxide. A toxic substance, hydrogen peroxide, is broken down by another peroxisomal enzyme into water and oxygen. They are abundantly found in cells that metabolize fats or lipids, such as liver cells. For example, alcohol is detoxified by peroxisomes in liver cells. Also, in germinating seeds, they convert fatty acids into sugars needed by the growing seedlings. And we have glyoxysomes, which are specialized peroxisomes in plants, which are responsible also for converting stored fats into sugars. Vesicles and vacuoles are membrane-bound sacs that function in storage and transport. Other than the fact that vacuoles are somewhat larger than vesicles, there is a very subtle distinction between them. The membrane of vesicles can fuse with either the plasma membrane or other membrane systems within cell, while the membrane of a vacuole does not fuse with the membranes of other cellular components. Again, the large membranous storage sacs in cells are called vacuoles. The smaller ones are known as vesicles. Vacuoles in animal cells vary in their uses. Some are for storage of water or food, while others are for excretion of waste materials. In plant cells, so you can see in the figure is actually an example of a typical plant cell. They have a very large central vacuole, the one in white. It's actually occupying up to 90% of the cell volume. As you can see, it's very large. It's called the large central vacuole. And they function for intracellular digestion, space filling, and control of cell turgor. Aside from water, sugar, and salts, vacuoles in plant cells also contain pigments such as anthocyanins and toxic molecules. The toxic molecules help plants from herbivorous animals. In some protists, contractile vacuoles rid the cell of excess water. Further, certain structures in the cell exist for very specific functions, such as the centrosomes. In animal cells and in some protists, the centrosome is found just outside the nucleus. It consists of two rod-shaped centrioles which are at right angle to each other. A centriole is made of bundle of microtubules, as depicted on the picture. You have the hollow tubes you see are microtubules forming up a centriole. When a cell is about to divide, the centrioles replicate, resulting in two pairs of centrioles. The two pairs of centrioles move apart, and each pair becomes part of the newly formed cells in cell division. The centrioles function in the formation of the spindle fibers, which help to pull chromosome pairs apart to opposing ends of the cell during cell division, which we will be discussing on the next lessons. Like the mitochondria, chloroplasts also have their own DNA and ribosomes, the one in green depicted in the picture. But chloroplasts have an entirely different function. Chloroplasts in plant cells carry out photosynthesis. Photosynthesis, by the way, is the series of reactions that use carbon dioxide, water, and light energy to make glucose as end product and oxygen as a byproduct. Like mitochondria, chloroplasts have outer and inner membranes. Within the space enclosed by a chloroplast inner membrane is a set of interconnected and stacked membrane sacs called thylakoids. Each stack of thylakoid is called granum. The fluid in the figure, the fluid enclosed by the inner membrane that surrounds the grana, or in singular form granum, is called the stroma. The chloroplasts contain a green pigment called chlorophyll. Chlorophyll captures the light energy that drives the reactions of photosynthesis. Like plant cells, photosynthetic protists also have chloroplasts. Some bacteria perform photosynthesis as well, but their chlorophyll is not relegated or assigned to an organelle. The cytomembrane system, also known as endomembrane system, refers to a network of pathways through which materials flow to the different parts of the cytoplasm. Also serves as a passage for the entry and exit of certain substances in the cell. Like the cell membrane, the membranes that make up the endomembrane system also consist of a phospholipid bilayer. This system of membrane pathways includes the ER, Golgi bodies, and some enzyme-filled sacs, and various transport vesicles. Part of the endomembrane system is the endoplasmic reticulum, which consists of flattened sheets, sacs, and tubes of membranes that extend throughout the cytoplasm of eukaryotic cells. It is structurally continuous with the nuclear membrane, and it specializes in the transport of lipids and membrane proteins. As you study the picture closely, it shows the two kinds of ER, or endoplasmic reticulum. You have the rough ER and smooth ER. The rough ER generally occurs as flattened sheets, studded on its outer surface with small spherical bodies called ribosomes that are engaged in protein synthesis. Ribosomes in the RER makes membrane proteins and proteins for export out of the cell. The proteins made by ribosomes in RER or rough endoplasmic reticulum are then threaded into the interior of the rough ER to be modified and transported. Without attached ribosomes, the SER is usually a site for fat metabolism and forms vesicles for transporting large molecules to other cell parts. Functions of the smooth ER specifically includes synthesis of carbohydrates, lipids, and steroid hormones, detoxification of medications and poisons, and storage of calcium ions. In muscle cells, a specialized SER called the sarcoplasmic reticulum is responsible for storage of the calcium ions that are needed to trigger the coordination contractions of the muscle cells. Named after Camillo Golgi, who discovered it in 1898, the Golgi apparatus is a system of flattened, membrane bound sacs that look like a stack of pancakes. It is involved in modifying, sorting, and packaging macromolecules for secretion or for delivery to other organelles. Associated with the apparatus are the rounded vesicles, as you can see on the upper part of the picture on the left. These rounded vesicles have been pinched off from the ends of the flattened sacs of the Golgi apparatus and are believed to be carriers of materials between the apparatus and other parts of the cell. The number of Golgi bodies in a cell depend on the function and stage of development of the cell. Numerous Golgi bodies are found in dividing plant cells and in enzyme secreting animal cells. We have already mentioned that vesicles can bud from the ER and transport their contents elsewhere. But where do vesicles go? Before reaching their final destination, the lipids or proteins within the transport vesicles still need to be sorted, packaged, and tagged so that they wind up in the right place. The sorting, tagging, packaging, and distribution of lipids and proteins take place in the Golgi apparatus. The receiving side of the Golgi apparatus is called the sys phase. That's on the upper portion in the figure. And the opposite is called the trans phase. The transport vesicles that formed from the ER travel to the sys phase, fuse with it, and empty their contents in the lumen of the Golgi apparatus. As proteins and lipids travel through the Golgi, they undergo further modifications that allow them to be sorted. The most frequent modification is the addition of short chains of sugar molecules. These newly modified proteins and lipids are then tagged with phosphate groups or other small molecules so that they can be routed to their proper destinations. The modified and tagged proteins are packaged into secretory vesicles that bud from the transphase of the Golgi. While some of these vesicles deposit their contents into other parts of the cell where they will be used, other secretory vesicles fuse with the plasma membrane and release their contents outside the cell. Another membrane-bound organelle is the lysosomes, which is also produced by the Golgi apparatus. Of various sizes and shapes, lysosomes are known as digestive sacs in the cell. They contain numerous hydrolytic or digestive enzymes for the breakdown of carbohydrates, proteins, and fats within the cell. They also play a role in the destruction or repair of defective parts of the cell. In the process known as phagocytosis or endocytosis, a section of the plasma membrane of the macrophage invaginates or folds in and engulfs large substances such as food or pathogens. The invaginated or folded section with a substance, or food or pathogen, pinches itself off from the plasma membrane and becomes a vesicle. The vesicle then fuses with a lysosome And the lysosome's hydrolytic enzymes digest and or destroys the engulfed material. The cytoskeleton is a network of interconnected protein filaments which extends throughout the cytoplasm. It maintains cell shape, secures organelles in specific positions, allows cytoplasm and vesicles to move within cell, and enables cellular organisms to move independently. There are three types of fibers within the cytoskeleton. We have the microfilaments, intermediate filaments, and microtubules. Microfilaments are also known as actin filaments because they are made up of actin monomers. Microfilaments also provide rigidity and shape of the cell. Intermediate filaments are made of several strands of fibrous proteins that are wound together, which main function is for structural support. They bear tension, thus maintaining the shape of the cell and anchor the nucleus and other organelles in place. The picture shows how intermediate filaments create supportive scaffolding inside the cell. Then we have the microtubules. As their name implies, microtubules are small, hollow tubes. They help the cell resist compression, provide a track along which vesicles move through the cell, and pull replicated chromosomes to opposite ends of a dividing cell. As you examine further, you find the control center of the cell, the nucleus. The nucleus is surrounded by a nuclear envelope. The nuclear envelope is a double membrane which contains nuclear pores for materials to enter and exit nucleus. The nuclear envelope is punctuated with pores that control the passage of ions, molecules, and RNA between the nucleoplasm and cytoplasm. The nucleoplasm is the semi solid fluid inside the nucleus where we find the chromatin and the nucleolus. The nucleus is generally an oval shaped or spherical shaped structure. The nucleus is known as the control center of the cell, where it regulates and coordinates all the activities of the cell. Inside the nucleus is the chromatin, which is composed of DNA and protein molecules. During the cell division, the chromatin coils and becomes condensed into rod shaped structures called chromosomes. The DNA found in these chromosomes make up the so-called genes, which are the bearers of hereditary traits. The DNA is a double helix structure with alternating sugar and phosphate group backbone with hydrogen bonded nitrogenous base pairs. We have the innermost part of the cell, specifically the innermost part of the nucleus, the nucleolus. The nucleus directs the synthesis of ribosomes, but how is it possible? The nucleolus is organized around regions of some chromosomes containing DNA that encode ribosomal RNA. The darkly stained area within the nucleus is the nucleolus, which aggregates the ribosomal RNA with ribosomal proteins to assemble the ribosomal subunits that are transported out through the pores in the nuclear envelope to the cytoplasm. The nucleolus is considered a ribosome production factory. Some other cells have variations in structure and functions. For example, specific cells have cilia and flagella. The flagella, or flagellum in singular form, are long tail-like structures that extend from the plasma membrane and are used to move an entire cell in a propeller-like fashion. Examples of cells with flagella includes the sperm and spirillum. Cilia, on the other hand, are hair-like structures that are used to move entire cells or substances along the outer surface of the cell. They are usually found in the cell's lining fallopian tube, lining of the respiratory tract that trap particulate matter and move it toward your nostrils, and in some protists and bacteria. As you get to examine all of those organelles, their structures and functions, We get to realize that cells carry out specific functions with the aid of non-overlapping yet communicating organelles. And the number of cell organelles, in addition, vary from one organism to the other as well as its shape. However, their functions are quite similar, and that is to carry out metabolic activities necessary to sustain and perpetuate life on Earth. If you want to keep updated on my next lesson videos, please subscribe to my channel. Thank you so much for watching.